Welcome to Biology Made Easy. In this lesson, we are discussing solute potential values in cells and sieve tubes. Well, what is water potential? I have this system. It contains only water molecules, so it's pure water, no solutes. Now, the tendency of this pure water to move into any system is the highest. So is the highest water potential. We discussed water potential in our lesson on water potential. Please check that so that this one will be easy. So this pure water has no solutes. And when we bring in molecules or solutes, what happens is that the water molecules, some surround the solute and separate the solute into ions or molecules, that is, dissociate the solute. In the process, the concentration of water or hydrogen ions decrease. So in this process, the solutes dissolve in the water, and so the water potential reduces. So the measure with which solutes dissolve pure water to reduce the concentration of the water molecules, reducing the water potential, is solute potential. So in this one, small amount of solutes, some of the water molecules will surround the solute molecules, and so they will lose kinetic energy. They will not be able to move fast, and a few water molecules will be there to move. So the solute lowers the water potential. And the measure by which it lowers the water potential is the solute potential. So solute potential is always measured in the negative. So the higher the solute potential, the higher the negative. So let's go on gently like this. This one, few molecules of solutes. So we have low water potential here and low solute potential. In the pascals, we have here pure water is 0 water potential. When the water potential is low, it means it's lower than 0. So it's also negative. And solute potential is also always negative. So this solute potential is low. So the negative is low. Now, what happens when we increase the solutes? So in this case, the solute amount is increased, so it will reduce water potential, and so water potential is low. And when water potential is low and is lower than here, it will have a much lower negative. And this higher solute potential will have more negative. The greater the solute potential, the higher or more the negative. So here is plenty of solutes and very low water potential, but very high solute potential. So the solute potential will be greatly higher negative. So that is solute potential values. The less solute in the system, the lower the solute potential, the less negative. The more solute in the system, the higher solute potential, it means more or great negative pascals. Well, let's look at it well also in here. In this system, we have 3 plant cells with different concentrations. This one, cell A, has few solute molecules and plenty of water molecules. So we have more water molecules here. We have high water potential. It's very high. It's more close to pure water. So you see the water potential is negative, but not so negative. You come here, less water molecules means low water potential, and it's more negative. This one has more water molecules. They have a high water potential. You see that this water potential is higher than this one. So the higher water potential means less negative. It's more towards zero than this one. Meanwhile, when you come to look at solute potential, this has less solute, system has less solute, so it has less negative. Here has more solute, so it has more negative than here. And here has less solute, so it has low solute potential. And you see that the solute potential here is lower than here. And so it has less pascals, less negative. So that is how to look at solute potential. It's always measured in the negative. We now want to look at what happens to solute potential in plant cells and sieve tubes. We first of all want to explain some terms. We have a term like solute, and the solute you find in sieve tubes, that's the phloem sap. The solute you find in sieve tubes is largely sucrose. Sucrose because photosynthesis takes place in the leaf and in the green parts of the plant, and photosynthesis will produce glucose. But why is it that glucose is not found in the cells but sucrose? Well, you know it, isn't it? Glucose is very reactive. Glucose can easily be used for respiration or turn into other metabolites. So sucrose is less reactive. And so the substances that are found in cells or sieve tubes that are moved around is largely sucrose, with little amounts of amino acids and ions and very little amounts of hormones. And these substances are called assimilates. So that's about solutes. Now, another term we want to talk about is source. The source is any place where sucrose is put or loaded into the phloem, that's the sieve tubes. So anywhere that sucrose will be put into the sieve tubes is the source, and it's usually in the leaf or dormant buds. Then we look at another term, sink, and the sink is any place where sucrose will be taken from the sieve tubes and used in cells. And sinks are usually roots, storage organs, fruits, and regions of growth. So sink, there are areas where sucrose is removed from the phloem. And when sucrose is removed, the sucrose will be turned into fructose and glucose and turned into other substances. So that is the sink. Now we have other terms to look at. We look at loading of sieve tubes. And loading simply means movement of sucrose from photosynthetic cells. You know, the cells in here will be photosynthesizing and making glucose, turning into sucrose. And the sucrose will be pushed into the sieve tubes and companion cells. That's the phloem. So any place that this goes on, that is the source. So the source is usually the loading points. Also here, another term, unloading sieve tubes. And unloading sieve tubes means areas where unloading sieve tubes means movement of sucrose out of sieve tubes into cells to be used. And unloading sieve tubes usually occurs at the sinks. Well, there's also another term, translocation. And translocation is simply movement of dissolved substances through conducting vessels. And the movement is from source to sink. Sucrose will move from the source to the sink. And sometimes sucrose also move from sink to other growing areas. And so translocation will occur between the sink and other growing areas, even growing areas that store food, like fruits. Let's note that when substances are moving from source to sink, they move in a different sieve tube. And when it's moving from sink to another place, they move in different sieve tube. The substances do not move in the same sieve tubes, different sieve tubes. Good. Now, having known these terms, we now want to look at solute potentials in the plants. So we are going to look at solute potentials from the leaves down to the roots. Well, here we have this drawing here. This is a section of the leaf. We have the petiole here, then you have the stem, you have the roots, we have the sieve tube, and some of the sieve tube down into the veinlets in the leaf. Now, we have labeled A as a palisade cell. It's a palisade cell here, A. Here, spongy cells, that's a B, spongy cell. And we have put in here C. We have said it's a sieve tube. Right. Now, before we put down the solute potentials values in pascals, we want to tell ourselves that the solute potential at the source in the leaf will be high, especially the solute potential in the palisade cells, where photosynthesis takes place greatly because the palisade cells have a lot of chloroplasts. So photosynthesis will be highest in the palisade cells, making glucose and being turned into sucrose, which is temporarily stored in vacuoles in the palisade cells. And this sucrose will move from the palisade cells into the spongy cells. So if you have palisade here, to move into the spongy cells and move into the veins, specifically into the sieve tubes and companion cells here. So let's come back here. We have the palisade cell, spongy, and a sieve tube in the veinlets. Now, the solute potential here will be highest because, as we have said, photosynthesis. Now, because the solute potential is high here, the solutes will move from cell A to cell B, that's a spongy cell, and the solutes move by diffusion through the apoplast pathway, the symplast pathway, vacuolar pathway. We have discussed these pathways on transports. So the solutes will move through these pathways from the palisade, where the solute potential is very high, into the spongy cells, and then move into the sieve tube and companion cells. And when the substances are moving into the sieve tube or companion cells, the substances can move by active transport and actively push the sucrose into the sieve tubes using energy from ATP. So the sucrose potential, the highest in the palisade cell, can be lower. That is why the diffusion can take place to move it into the spongy cells, but it can be highest still in the sieve tubes because of actively loading the sieve tubes. So let us look at the Pascal values that we will put all this to. Pascal values of solute potential. Now, here we have our diagram again, and now we have put down points on the plant and label this point as the D here, the stem, E, point F, point G, point H, which we say is in the root, that's the sink. You know the leaf area is the source. And as we have said, the palisade cell is making the sucrose by photosynthesis, and so the highest solute potential we expect to be in the palisade cells. So we give it minus 15,000 pascals. So the solute potential in A is higher, in B is lower, so solute will move by diffusion, also by active transport. Then the solute is pushed into the sieve tubes greatly by active transport. So the solute potential in the sieve tube may be even higher than in the palisade cells. So we have imagined and given the solute potential in the sieve tube the highest and said, well, 30,000 pascals. So see here is the highest solute potential. Then as we go down, the solute potential decreases because the solute is being removed from the sink. Solute moved out into the cells to be used. So you see, the solute potential will be higher and decreasing down as you get into the root, that is the sink. So this is how solute potentials are worked out in plants. Well, if you found the lessons interesting and you understand it, please give us a thumb and subscribe. Thank you very much and goodbye.